Okay, so we'll now continue our lesson. This time we're talking about microbial taxonomy. It's our lesson number 2. So microorganisms have different variety, have different kinds of microorganisms. So our course is medical or clinical bacteriology, but just so to make a point today, when you say microorganisms, we are not just referring to bacteria, but we are also referring to other microbes as well, such as parasites, fungi, and viruses. So bacteria are actually prokaryotes. Okay, so again, bacteria would be the plural form, and then bacterium is the singular form. Okay, so bacteria are examples of organisms with primitive nucleus. Okay, so in fact, the term prokaryotes, okay, ang ibig sabihin ng prokaryotes, pro means before, and karyo refers to nucleus. And the term prokaryotes refers to primitive nucleus because they do not have nucleus at all. Okay, they do not have nucleus at all, but it means that they don't have nuclear membrane. No nuclear membrane. So that's what we meant. That's what we meant by primitive nucleus. No nuclear membrane. Okay. So unlike eukaryotes, humans such as us, we are eukaryotes. Our nucleus. So instead of calling it as nucleus, prokaryote, we simply call it as nucleoid. So they do not have organelles, and of course, they are unicellular, meaning to say, when you say unicellular, means that bacteria are considered to be as the single-celled organisms. So prokaryotes such as bacteria, okay, are majority of their divisions are gram-positive and gram-negative bacteria. And what is the basis of gram stain? Gram stain is the basic stain that we're using. It's a type of differential stain. And we can divide it whether they are gram positive or gram negative. And what is the basis? What is the basis of gram staining? So the basis of gram staining would be the cell wall. Okay, would be the cell wall, particularly the peptidoglycan. As you can see, um, gram-positive bacteria, gram-positive bacteria, this is the gram-positive bacteria, and this would be the gram-negative bacteria. Gram-positive bacteria would have a thicker peptidoglycan. So the peptidoglycan is thicker, and the peptidoglycan is made up of teichoic acid. Whereas the gram-negative bacteria would have a thinner peptidoglycan, but instead of teichoic acid, they have the LPS or the lipopolysaccharide. Okay, so that is for the gram-negative bacteria. Okay. Now, parasites. You have already studied parasitology when you were in second year. And of course, parasitology is an example of eukaryote. So when you say eukaryote, it means that they have the true nucleus. They have a nucleus, organelles are present. So true nucleus, they also have organelles. So like humans, okay, so parasites are eukaryotic. So they can either be single or multicellular. So for example, amoeba, such as protozoan, such as amoeba, they are single-celled organisms. Whereas tapeworms, hell means they are multicellular organisms. Okay, so parasites could either be motile or non-motile. Okay, so protozoans are categorized by flagella. So, for example, flagellates as Giardia, they are flagellated. Amoeba have pseudopodia, Balantidium coli, such as Habsilia. So, malaria do not have an obvious means of locomotions. Okay, so these are the parasites. I will not really dwell into this one. And moving on to fungi. So we have a separate course for this one. Next sem, uh, after passing bacteriology, so the next course would be mycology. So mycology is an example of eukaryotic organism. Okay. So um, mycology is the study of fungi. So fungi are heterotrophic eukaryotes. So when you say heterotrophic eukaryotes, they obtain nutrients through absorption. So they obtain nutrients through absorption. And of course, um, most of these nutrients are actually would actually come from decaying matters because fungi, the most important characteristic of fungi is that they are saprophytes. So when you say saprophytes, it means that fungi live on decaying organic matter. Okay, so that's what we meant by saprophytes. So most of these fungi will live on decaying organic matter. So fungi could also be unicellular or multicellular. Yeast. So remember Saccharomyces cerevisiae, the one that we are using for fermentation of wine. So yeast is an example of unicellular microorganisms. So yeast is an example of unicellular microorganisms. But aside from yeast, I'll give you another example. Candida albicans. Okay, so Candida albicans is another example of yeast, and they are unicellular microorganisms. Okay, fungi, such as molds, are multicellular fungi. Okay, so some fungi exhibit dimorphic form. So what does it mean? So when you say dimorphic form, it means that some fungi would have two morphologic forms. So some fungi, such as, let me give you an example, Histoplasma capsulatum. Histoplasma capsulatum, when you expose them at 37 degrees centigrade, which is uh, at body temperature, they became yeast. They become yeast. But if you expose them at room temperature, approximately at 25 degrees centigrade, then they become filamentous. Or when you say filamentous, they are now molds. So they exhibit two morphologic forms. And since they exhibit two morphologic forms, that is the reason why we call them dimorphic. So the term di means two. Morph means morphologic forms. Okay, so dimorphic fungi. So such as Histoplasma capsulatum. Okay, so eukaryotic organisms would have nuclear membrane. Okay, so they have nuclear membrane such as this, and they also have organelles. They also have organelles. Unlike bacteria, they do not have nuclear membrane. So it's safe to say that do not have bacteria do not have nucleus. They just have nucleoid because they do not have nuclear membrane. Okay, so now let's talk about viruses. Viruses are another types of microorganisms, but this time viruses are considered to be as the smallest, one of the smallest infectious particles, but it's not the smallest. No, the smallest are the prions. Okay, viruses are smaller than what is seen by light microscopes. In fact, you cannot use you cannot use light microscope in order for us to visualize viruses. We have to use electron. We have to use an electron microscope. You have to use an electron microscope, okay, in order for us to see viruses. So viruses are known as the filterable agents. So viruses are known as filterable agents. Why? Because they are so small that they can even pass through. They're smaller than bacteria, which means that they're able to pass through bacteriologic filter. Okay, they're able to pass through bacteriologic filter because they are even smaller than bacteria. Okay, so viruses may contain either DNA or RNA. So they cannot possess two nucleic acids. Bacteria can have DNA or RNA. Bacteria can have DNA and RNA at the same time. Humans such as fungi, protozoans, parasites, all of us can have DNA and RNA at the same time. But for viruses, they only have they would only uh, they would only have one kind of nucleic acid, either a DNA or RNA. Let me give you an example. SARS-CoV-2, the causative agent of COVID-19, is an example of RNA virus. Hepatitis B virus, the one that can cause hepatitis B, is an example of DNA virus. Varicella, the one that can cause chickenpox, is an example of DNA virus. Rhinovirus, the one that can cause common colds, okay, is an example of RNA virus. Okay, so viruses are acellular. What does it mean? When you say cellular, they are not composed of cells. In fact, viruses are considered to be as a non-biological entity. So viruses are considered to be as non-biological entity, which means that they are non-living things. Okay, though they are not living things, viruses are still infectious. Okay, so viruses are obligate intracellular parasite. What does it mean? They would require live host cells. So they would require live host cells in order for us to cultivate them. What are examples of live host cells, such as the chick embryo? Such as the chick embryo. What else? The HeLa cells, the cells coming from Henrietta Lacks. We call that HeLa cells. Or the monkey kidney cells. The monkey kidney cells are examples of live cells. And we can use these live cells to cultivate viruses. So they are even more complicated to culture as compared to bacteria. Okay. Viral emergence become better known by DNA or RNA makeup. Like what happened in today's pandemic. That's an example of viral emergence. And it can affect host disease signs and symptoms, the chemical makeup of the virus. That's why we have so many variants, Delta variant, Omicron variant, and then it also affects geographic distribution. So, for example, monkeypox used to be known in Africa only, but now it's being distributed all over the world. COVID-19, the SARS-CoV-2, used to be confined in China, but now it is being distributed all over the world. Okay? Now, some of the viruses are resistant to lipid solvents and detergents. Okay? Particularly for non-enveloped viruses. Okay? So they are also resistant to pH and temperature changes. Now, the viral proteins, the viral proteins is the basis for the viral antigenicity. Okay? So if we are doing antigen tests, it means that we're trying to detect viral proteins from the human samples. Okay? So that is the antigenicity. And it's also the basis of serological methods such as the antigen test. Okay, so let's now discuss classification or taxonomy. Okay, so um, taxonomy is the systematic way or the orderly group of organisms to taxa or categories. And this was actually introduced by Carl Linné. So the one that you can see in the photograph, that's Carl Linné. Carl Linné, but it is spelled like this. Carl Linné is the father of taxonomy because he actually systematically classified all living organisms. Okay, and three structured interrelated categories. So based on this one, we can now have a systematic classification, that's taxonomy, nomenclature, and identification. So when you say nomenclature, it's a scientific way of naming microorganisms. So taxonomy is based on similarities and differences. Imagine during the time of Carl Linné, they are just basing with the morphometrics or phenotypic characteristics. Because during that time, there's no PCR, there's no there wasn't any science known as molecular biology. So it's kind of difficult for them to really systematically classify microorganisms. But because of the modern time, we now have the genotype or the genetic makeup. So this will now be known as the genotypic characteristics. Because of the genotypic characteristics, we can now properly classify and categorize living organisms. And this particular genotypic characteristics would be based on the DNA or RNA sequences, the comparison of the base composition ratio to measure the degree of relatedness of two organisms. Example would be the GC ratio, or the ratio between the guanine and cytosine in the nucleic acid of these particular organisms. So it is now actually a more accurate way of classifying the taxonomical categories of these particular microorganisms. So before the advent of molecular biology, wala pang genotypic characteristics, majority of taxonomies would only be basing on the phenotypic characteristics, not the genotypic characteristics. What do you mean by phenotypic characteristics? In phenotypic characteristics, we are trying to describe the phenotype. Okay, we're trying to describe the phenotype. And when we say phenotype, we are referring to the observable, those that we can manifest. Okay, observable physical and functional features of an organisms expressed by genotype. So before molecular biology, we were just basing it on the macroscopic and microscopic morphology, staining characteristic, nutritional requirements, antigenic markers, physiological and biochemical characteristics, susceptibility or resistance to antibiotics or chemicals. These are all known as phenotypic characteristics. Okay, phenotypic characteristics. So, uh, yan. Because of that, because of this uh, classification, we can now actually classify microorganisms into three domains. Okay, we now have the three domains. Okay, so these three domains was actually postulated by Carl Woese. So that's the reason why these three domains are sometimes known as the Woese classifications. And according to Carl Woese, there are actually three main domains. So the first domain is the domain bacteria. The second domain is the eukarya or eukaryota. And somewhere in between bacteria and eukaryota is the archaea, archaeobacteria. Okay? So the archaeobacteria are somehow interrelated to eukaryota and interrelated to bacteria. But those that are, if you know, this is how you interpret phylogenetic tree. If this particular organisms is far from these organisms, so what do you call this one? They are distantly related. But if you're going to look at Microsporidia and Diplomonads, they are close in phylogenetic tree. So how do we describe this one? They are closely related. So we can say that Archaeobacteria is somehow closely related to bacteria, but bacteria are distantly related to eukaryota. That's how we interpret, you know, phylogenetic tree in bird's eye view. Okay, so moving further, for viruses, it's quite difficult to classify them in Carl Woese domains. Why? Viruses are non-living things. So we cannot identify viruses in archaea, in eukaryota, or in bacteria. Okay? So viral classification is based on the genome, the replication, and the virion, and there is a separate committee in classifying viruses, and we call it the ICTV, or the International Committee on Taxonomy of Viruses. So the ones responsible in classifying viruses. Okay? So, but since we are talking about bacteriology, so this is how we will be classifying the bacterial classification. So first, we classify them according to domain, and then we classify them according to kingdom, and then division, class, and then order. Okay? For order, after the order, we will now be classifying them according to family. Okay? And how would you know if we're talking already about family? You'll be able to see the suffix "asi". So this is how we pronounce it, "asi". So for example, Enterobacteria cI, Enterobacteriaceae, Entero, Enterobacteriaceae, and then that's one example of bacterial family. And Enterobacteriaceae is actually divided into several tribes. And then the tribes are further divided into genus. And in writing scientific name, the genus is always capitalized. And the plural of genus is genera. And then after the genus, we now have the species. And they are always written in lowercase. Now remember, there's no such thing as species. Never say species. It is always with letter S, species. Because the singular form is written this one, and the plural form is written this one. Subspecies are being written if it's appropriate, but not all scientific names would have the subspecies. So let me give you an example. Escherichia coli. Escherichia is the genus, while coli is the species. And when we say species, okay, we are referring to a specific epithet. So we are referring to specific epithet. So that's what we meant by species. Okay? So the scientific name is the universal name of a certain living thing. For example, we are known as Homo sapiens all over the world because that is the scientific name of humans. Okay, so moving on. Yan, so as you can see, so Escherichia coli. So Escherichia here is the genus, while coli is the specific epithet or species. Now, in writing scientific name, you have to underline the genus and underline it separately with that of the species. However, if you're typing it, then you can have them italicized. And if they are italicized, there's no need to underline them. Also, if you have already mentioned Escherichia coli in one paragraph or in one document or in one chapter, the next time around, you may not mention Escherichia coli completely. You can use the abbreviated form, and the abbreviated form of Escherichia coli is E. coli. However, you cannot use E. coli right away without mentioning first Escherichia, because there are even some amoeba that is also abbreviated as E. coli. Remember, Entamoeba coli is also abbreviated with E. coli. So if you haven't mentioned yet Escherichia, then you cannot say E. coli because that would actually lead to confusion. Are you referring to Entamoeba coli or are you referring to Escherichia coli? So that's the reason why you have to completely express first the genus before abbreviating it. I hope that's clear. So the nomenclature provides assignments for each organisms. Okay, so remember, Aci refers to the family name, and the genus is always capitalized, and the species or the specific epithet always begins in lowercase letter. So genus or species, some genera have the same first letter, so the first syllable is used. So for example, Staphylococcus, Streptococcus. So it's better that we use the first syllable. So again, to avoid confusion, Escherichia coli and Entamoeba coli should not be confused. And one way of avoiding the confusion is to use the syllable instead of simply using this one. Okay, some organisms would have subspecies because of the phenotypic differences. Not only that, some of them would even have the serovarieties because of the serological differences. Some of them would even have biovarieties because of the biochemical test results differences. And now, a more stringent way of determining their differences is through phage typing. Because of it is now, this one is based on susceptibility to specific bacterial phages. What are bacterial phages? Bacterial phages are known as the bacteriophages. And bacteriophages are actually bacteria eating virus. These are viruses that can actually eat bacteria. Imagine that. Okay. Now, some bacteria would even be classified according to strains. And when we say strain, our primary basis is based on its susceptibility or resistance to a certain antibiotics. Example, penicillin. So there are strains that are susceptible to penicillin, and there are even strains that are not susceptible or resistant to penicillin. So that's what we meant by strains. Okay. So the in classifying the in determining the phenotypic or genotypic characteristic, genetic relatedness is very important. So that's the reason why in when you do PCR, the primer that you will be using are actually the 16S rRNA for prokaryotic organisms. So these are the primers in PCR. And if you're using um, eukaryotic organisms, then your primer should have been the X18S rRNA origin. Okay, so a while ago, I was talking about the Carl Woese classifications. And according to Carl Woese, there are actually three domains. Okay, there are actually three domains. So the first domain is the bacteria, and then the second domain is the eukarya, and the third one is the archaebacteria. In our lesson here in bacteriology, we will actually be concentrating on the bacteriology. But let me discuss some of the archaea. Archaebacteria are actually not being encountered routinely in clinical microbiology. And archaea would have three major classifications. So the first classification is the extremely thermophilic organisms. When you say extremely thermophilic organisms, they live at a very high temperature, in environment, very high temperature. And then we have the extremely halophilic organisms. When you say extremely halophilic organisms, these organisms live at, at a temperature with high salt concentration. And the third one would be the methanogens. And when you say methanogens, this particular archaea live in environment with a very high methane gas, such as in wetlands. Okay, so these are examples of the archaeobacteria. And the archaeobacteria is very important because the enzymes, the enzymes that we are using from PCR, actually the Taq polymerase. This one is the most important enzyme in PCR, and we actually get it from Thermus aquaticus, an example of thermophilic organism, Thermus aquaticus. Okay, that's the origin of the Taq polymerase. Okay, so, but according to cellular types, there are two major kingdoms. So we have the prokaryotes and then the eukaryotes. Okay? So prokaryotes and eukaryotes. So both bacteria and archaea are considered to be as prokaryotes. Okay? So when you say prokaryotes, means that there are no there's no nuclear membrane. Eukaryotes means they are provided with nuclear membrane. Okay. Now, what do you mean by pathogenic organisms? Pathogenic bacteria are prokaryotes that can infect eukaryotic hosts, such as humans. So we can be infected with pathogenic bacteria. Okay? So archaebacteria is most closely related to eukaryotic cells. And they contain microorganisms that can grow under extreme environmental conditions. Like I mentioned this to you a while ago, we have the extremely thermophilic bacteria. We have the extremely halophilic bacteria that lives on high salt concentration. And we have the methanogens that lives in that can produce methane gas. Okay? So archaebacteria do not have peptidoglycan. Unlike the true bacteria, they have peptidoglycan. However, they can stain gram-positive or gram-negative. So the cell envelope and enzymes present in archaebacteria allow these particular organisms to survive in harsh conditions, such as extremely thermophilic conditions or in extremely halophilic conditions. Okay, so that's about it. Okay, so we will just continue our lessons next time.